Good morning. Today we are going to dive deeper into cellular processing in the visual system. So, got my slides up here. So, we're going to go back to the network of cells that live on the retina. And each of these cells have different functions. And again, we have the sensory receptors, which are the rods and the cones, and they function differently based on them having different photopigments, which means that they're maximally responsive to different wavelengths of light energy. And their function is informed by their degree of convergence. So the rod system is a more convergent system than the cone system. The rod system makes more connections with bipolar and ganglion cells than the cones do, right? There are more rods that synapse onto a single bipolar cell to a ganglion cell, whereas you have some cones in the visual system that have this direct line to the brain: cone, bipolar cell, ganglion cell. Now, the sensory receptors synapse onto bipolar cells, and you can see in this figure here that mediating those connections are what's called horizontal cells. So horizontal cells communicate laterally across the retina, and again, they are going to moderate the messages that are sent to bipolar cells by the receptor cells. And we'll get to that in a minute. That's pretty much the basis of lateral inhibition. Then we have this other layer of cells called amacrine cells, which also communicate horizontally. They play a role in movement detection, shape detection, and then we have our ganglion cells. The ganglion cells are receiving the messages from the receptors through the bipolar cells, and their axons form the optic nerve, which travels out the back of the eye. That point in which the optic nerve exits the eye, there are no receptors in that part of the retina, and it's called the blind spot, right? So if there aren't detectors to detect information from outside, from out there in the world, there's going to be no messages from that area sent to the brain. So the brain fills in the missing information based on information from the other eye, but also from the cells that are surrounding the blind spot. It's called completion, and we talked about that last time. So these two slides are just written out what I was just saying when we were looking at that figure. So again, rods and cones make synaptic contact with horizontal and bipolar cells. The horizontal cells are going to send inhibitory messages to those bipolar cells when they're stimulated via the rods and cones. Bipolar cells make synapses onto amacrine cells and ganglion cells, and these cells have their various functions. Again, the ganglion cells form the optic nerve. Some information is going to cross over via the optic chiasm. Half of the axons from each eye cross over, so that information from the right side of the world is processed in the left hemisphere or the left occipital lobe, and information from the left side of the world is going to be processed in the right occipital lobe or the right hemisphere. Now, most of ganglion cell axons go to the lateral geniculate nucleus, which is a portion of the thalamus, right? Again, the thalamus is a sensory relay station. It's gonna send information to the primary processing area of the cortex. If we're talking about visual information, it's going to go to area V1 in the occipital lobe. But again, other sensory information goes to different parts of the thalamus, and then the thalamus relays that information to the proper processing area. So the LGN, lateral geniculate nucleus, is a portion of the thalamus, the sensory relay station. Now, that's where most ganglion cell axons go. Smaller amount goes to the superior colliculus, which is an area involved in integrating visual and auditory information. And then some axons go to other areas of the brain. So when I ask you the pathway for processing visual information, I'm talking about the network of cells in the retina to the LGN to the primary processing area in the occipital lobe. Okay. What I didn't say already on this slide is that the cortex and the thalamus are in feedback. They're feeding information back and forth constantly. And actually, the thalamus receives more information from the cortex than it sends. Our top-down processing, that function of completion, is somewhat informed by this reciprocal communication. Here's a figure so that you can see the pathway for visual information. Okay, so I'm going to show you some pretty neat illusions or perceptual phenomenon, and we're going to talk about why it is that these illusions happen and what they're an example of, that it's important for us to process information in this way out in the real world. So this is called the Chevreul illusion. Oftentimes you'll hear it as the staircase illusion. And what you should be seeing at the boundaries of each color change is this increase in brightness on one side and a little dark band on the other. I don't know if you can see that, but again, there's a little dark band right here closer to the darker color and a intensity change right here, and it's a bump up in brightness. And you see something like that at each variation in the intensity of the color. This is called the Hermann grid, and if you look at this long enough, you probably see grayish circles at all of these intersections. Okay. Now, if you measure the physical properties of this stimulus, if you measure the nanometers of waves or the wavelength of light, the intensity of the light that's being reflected into your eye, there is no change in the physical property of the stimulus at these intersections. This is a function of our nervous system. That's our perception. Same with the Chevreul illusion, right? There's no change in actual physical intensity when you see these bumps up in brightness and decreases in brightness. So when you look at the chevron illusion, again, this figure is talking about the intensity of the light information not changing from A to B. Right? From A to B, there's no change in intensity. Then from B to C, there is a change in light wave properties, but there's no change from C to D. Your perceptual experience does not match this curve, this figure of your light intensity. Right? Your perceptual experience looks more like what's captured in this figure, right? When you go from A to B, once B changes into C, you have this bump up in brightness. All right, that's that bar that's kind of running down this border right here that you can see. It looks a little more, it looks lighter, a little more intense. And then you see this dip right here is capturing this dark bar that you're seeing right at that boundary. And then again, there we have going from C to D, which there is no change in light intensity from this point to this point, from C to D. Okay. So this is your perceptual experience of the chevron illusion, and again, that does not match the physical change of light wave information for this example. So lateral inhibition is the name for the cellular processing that happens in the retina. And it's important because what it does, it's going to sharpen the contrast to emphasize the border of objects. Okay, so again, when there's this change in light wave intensity or from one color to the other, there's this bump in intensity or dip in intensity to sharpen the contrast, right? It allows us to see the boundaries of objects. And lateral inhibition is the reduction of activity in one neuron by activating neighboring neurons, right? So if one receptor is stimulated, what that's going to do is stimulate a horizontal cell that is going to send an inhibitory message to the neighboring bipolar cell. Now, the responses of the cells in the visual system depend upon a net result of excitatory information and inhibitory information. So again, lateral inhibition, or this sharpening, the sending of inhibitory messages to neighboring cells is going to sharpen the contrast at the boundaries or borders of objects. And this happens at the level of the horizontal and bipolar cells. Okay, so here we have a figure that hopefully describes this a little clearer, okay? Let's say that, you know, receptors hold on one sec. Okay, so the image, area A and B, that image is reflecting light that's striking cells in your retina at a high intensity, which makes it look lighter than C and D as part of this illusion, right? That is bouncing off and reflecting into your eye lower intensity light, okay? So if we look at our top figure here where it says high intensity and low intensity, we have receptors that are being stimulated at points A and B, C and D, and all of those have neighboring cells, okay? Now, for the high intensity light, that's about 100 units of energy. And again, if that cell is stimulated, it's sending 100 units of energy. Its stimulation of horizontal cells is going to send a bit of inhibitory information to its neighbors. Let's say it's about a tenth of that information is, or that energy is inhibitory, and it's being sent to its neighbor, okay? So what that means is that, you know, cell A has a neighbor that's sending it a tenth of its energy in inhibitory messages, and cell A is receiving a tenth of inhibition from its neighbor X and its neighbor B, and it's sending 80 units of energy to its bipolar cells, okay? Cell B is receiving 10 units of inhibitory messages from cell A, but it's only receiving 2 units of inhibitory energy from its neighbor, because its neighbor is receiving a lower intensity light, right? Its neighbor only has 20 units of energy to send as opposed to 100 units of input to send. So the net response of cell B, it's receiving more inhibitory energy from one cell to the other, and it's getting less inhibitory energy than A, is that its net response is higher, and it causes that bump or that increase in intensity of light. Okay? That's accounting for this light band or bump up in brightness that's happening at the changeover from B to C. Now, because cell B is one of C's neighbors and it's under high intensity, it's receiving 100 units of energy. It is going to send a tenth of its energy to its neighbor in inhibitory messages, so that's -10. Cell C's neighbor D is receiving lower intensity light, so its output is at 20 units, so a tenth of that is -2. So cell C's net response is much lower than cell B's and also lower than cell D's. So that accounts for that dark border that's running down here, okay? Now you can see here for cell D, that's receiving -2 from both of its neighbors, so it's at 16, and so is its neighbor Y, okay? So again, if you look here from point A, then this increase in intensity to 88, this big dip or decrease in intensity, and then these are at 16, accounts for the perceptual change from A to B, the brightness boundary, the dark border, and then the same intensity from C to D. Okay. So if we chart the energy that's being sent to bipolar cells, given the figure that we were just looking at, our perceptual experience, our graph or chart of our perceptual experience, is a match to the responding of the bipolar cells when receiving inhibitory messages from their neighbors. So lateral inhibition does a really nice job at explaining this type of phenomenon. And the same thing happens with the Hermann grid, right? Here we have a situation where A would be the cell that's under point A in the grid, and this cell is receiving all these units of inhibitory messages from its neighbors, a 10th coming from here, a 10th coming from here, here, and here, it's going to send less excitatory messages to the bipolar cells, which are next in the chain, and it's going to make it so that this portion of our world is going to look dimmer than the surrounding areas. So again, lateral inhibition or receiving inhibitory messages through horizontal cells does a nice job of explaining our perceptual experience of these illusions. Now, the question you may be asking is why is this important, right? How does this impact our experience in the real world? Well, it has to do with contrast sensitivity, right? So remember when you watched the TED Talk and it took a while for you to be able to spot that predator in the wooded area? Part of what's responsible for that is color. Color can create contrast, but it's being able to separate an object from the background or a figure from the ground, okay? And again, lateral inhibition will sharpen the boundaries or the edges of a particular object, which has it stand out from the background once those features are combined, right? So this is, on your right, is what the world would look like without contrast sensitivity, okay? You wouldn't be able to discriminate an object from the background. You wouldn't be able to see the borders or the boundaries of objects. Okay. Here's just an example. This is strong contrast. And the less of a difference between the object and the background, the much harder it becomes to separate objects from the background. Okay. So if we're going to talk about cellular processing, we have to talk about a concept called receptive fields. And the receptive field is the part of the visual field that either excites or inhibits a cell in the visual system of the brain, right? So the receptive field is just that point in space where light is coming from that strikes the receptor, okay? And when you move further along in the pathway of processing visual information, receptive fields start to become more complex, and they become made up of the receptive fields of other cells through convergence. I put this great video up here on YouTube. Please watch it. It's great at explaining ganglion receptive fields. Right, so this is what I was just saying, right? For your rods and cones, cells in the retina, the receptive field is the point in space in which light is coming in and striking that retina, that receptive cell in the retina. When you move along in the pathway for processing visual information, receptive fields are derived from the visual fields of particular cells. Right? Ganglion cells, for example, converge to form the receptive fields of the next level of cells. So here, right, here's the eyeball, and lining the back of the eye is the retina. And the retina is lined with receptors, right? So this point in space, let's say there's an object out here reflecting light into the eye, okay? It's part of the visual field. The visual field is everything that you can see, right? It's your whole area of view. And this point in space has an object that's reflecting light into the eye that strikes receptors on the retina, right? This point in space is this receptor's receptive field. It's receptive to light from that point in space. Now again, when we get to cells further along in the pathway of processing visual information, especially when there is convergence, right? Now all of these receptors are part of this cell's receptive field, okay? These cells are all converging onto this cell here. So anything out in the world that's reflecting light and hitting any of these cells, any of these receptors are part of this cell's receptive field. Again, I would watch that video. It's extremely helpful in describing receptive fields. Okay. Now the receptive fields of ganglion cells, right? Receptors, bipolar ganglion cells are in 3 categories. Parvocellular neurons, so the cells that feed the information to this ganglion cells are mostly located in or near the fovea. They have smaller cell bodies and smaller receptive fields. They're highly sensitive to detect color and visual detail. Okay. Magnocellular neurons, distributed evenly throughout the retina, have larger cell bodies and visual fields, are highly sensitive to overall patterns and movement. Okay. Coniocellular neurons have small cell bodies found throughout the retina and have several functions. Their axons terminate in multiple different places. Now, the ganglion cells that were fed by receptors, bipolar cells, ganglion cells, they send information through the optic nerve to the thalamus, to the LGN of the thalamus. And the cells of LGN have similar receptive fields to ganglion cells. They have what's called center surround receptive fields. So there's an excitatory portion and there's an inhibitory portion, and that is surrounded by either an excitatory or inhibitory section as well. I'll show you an example of that in one moment. I just want to reiterate here that the LGN is the sensory relay station. Okay, it regulates neural information from the retina to the visual cortex. It only sends a particular amount of that information to the cortex. Okay, so here we see center surround. They're called on-off center surround receptive fields. And again, they can come in 2 flavors. And they have opposing centers and surrounds. For this cell, the center is excitatory and the surround is inhibitory. And here you have the opposite pattern. Okay, and so how this cell responds to input, okay? Because this is an excitatory center, inhibitory surround receptive field cell, if the light information is activating or striking the center of this cell, there's only a little bit, right, this small dot of light, that cell's going to respond, but the timing of the response is going to be somewhat slow, right? Let's say we increase that spot of light So it is exciting the whole area of the center, okay? Because the center is excited and there's light activating that area or stimulating that area, the timing of that neuronal response increases, right? You see that cell is firing faster, right? Again, information is about timing and patterns of responses in the cells. Now, if we move to C and we increase the light information, it's starting to move or bleed over into the inhibitory portion of this cell. So it's going to slow the firing of that cell down. Again, cell response is a net result of excitatory and inhibitory information, right? So the firing of this cell slows down, and once the light is covering or the stimulus is activating this whole receptive field, it slows down significantly. Okay. So here we have an example of a center surround receptive field, right? There's light being reflected off of something in the world. It is hitting these receptors, right? This part is going to be the on section for this ganglion cell's receptive field, and this is going to be for the inhibitory section, right? So if there's light information shown across this whole area, this is going to have a slow firing response. Okay, so again, you have the network of cells in the retina, right? Ganglion cells form the optic nerve. The optic nerve sends most of its information to the lateral geniculate nucleus, and from there, the information is going to be sent to the primary visual receiving area in the occipital lobe. The occipital lobe is called the striate cortex based on its appearance. This area is also called V1. Okay, so the primary processing area for visual information is V1, and it is the site of the incoming information from the LGN, and it is the surface of the cortex. From there, information goes to other receiving areas, and there's also two pathways, one to the temporal lobe and one to the parietal lobe, and finally arriving at the frontal lobe. And so we'll get to these pathways in a moment. In a moment. So again, here we have V1, the primary area for receiving visual information from the thalamus. Right here we have area V2, the secondary receiving area, area V3. Then we have our streams, ventral stream going to the temporal lobe, ventral stream going to the parietal lobe. I hope I said dorsal stream. Right, going to the parietal lobe. Here we have area V4. V4 is important for processing color information. And here we have area V5 or MT, medial temporal, that is responsible or plays a role in processing movement information. Okay. So given that we have these different areas, and we're going to talk about some more, like the inferior temporal cortex is going to be important for some other things we're going to talk about in a moment. We talk about the brain as being modular. modular by function, right? There are different areas that are primarily responsible for processing certain types of information. But there's also what's called distributed representation, right? So there's you know, it's not just area V4 that's responsible for color perception. It's going to also be fed through activation in multiple areas of the brain. Okay. Oh, and we did talk about how the cortex has columns. It's organized in columns and layers. These are examples of the layers for the visual system. Now, it also has a columnar organization. Right? Cells within given columns process similar information. Maybe it is the direction or angle of a light information in the world. In a column, that column will process that angularity of light information. When we move into the cortex, okay, into area V1 and beyond, neurons begin to fire to more complex features and more specific features. Cells in the visual cortex are called feature detectors, and we have cells in V1 that are simple cortical cells. They have side-by-side receptive fields. They're very similar to our on-off center surround, but instead we have an on-off side-by-side receptive field. And they respond to orientations in the environment. We also see complex cortical cells which respond to moving bars of light in particular orientations. Okay, so moving bars of light as opposed to stationary bars of light. And stop cells respond to corners or angles of bars of light in a particular length. So here's an example of a simple cell, okay? Again, we have this excitatory area of its receptive field, right? So if light is shown in this area, there'll be neuronal response. The more the light fills up this area in the center, it's excitatory, the more rapid the firing will be. If that light bleeds into these inhibitory sections, that firing rate will slow. Okay. Now, again, you'll have a column of cells that are going to respond to orientations of light. All right, so here's an example of this side-by-side on-off receptive field simple cell. Again, like I was just saying, if the light is filling up this excitatory area, you'll see this firing rate. If that light out in the world is at a different orientation, right? So it's bleeding over into these inhibitory areas, it'll slow down the firing of this type of cell, okay? Now, the visual system not only has cells, simple cells that respond to this orientation, right? It has cells that like bars of light at 45-degree angles, 75-degree angles. So there are cells in the visual system that are going to be maximally responsive to this type of orientation and would actually be inhibited by this type of orientation. And you can find these cells in columns within the cortex. Okay, complex cells. Complex cells are located in V1 or V2. V2 is the secondary processing area. And again, they're responsive to bars of light in a particular orientation, but they respond most strongly to moving stimuli. End-stop cells, again, they're similar to complex cells, but they have a strong inhibitory area at one end of its bar-shaped receptive field. So here is a nice chart that describes the characteristics of the receptive fields and what type of cell they are. For your exam, I would focus on this figure. This one is just a different way of delivering that kind of information. It also tells you more cleanly where these cells are located. Okay, so there are cells that respond to some patterns of light and not by others. As we just saw, there are some cells that will respond to orientations that are vertical. There are some cells that like horizontal or diagonal orientation. Again, there are some cells that are going to respond to light that moves or light that or light in angles with movement. And so as you saw, we were going from, center surround to these side by side, to responding to movement, to responding to edges and angles. As we move along in the pathway for processing information, neurons are going to fire to more complex stimuli. Okay, so we have area V2, secondary visual area, that receives information from V1. V1 and V2 are going to send information back and forth to one another. There's also this ventral stream, a pathway that goes through the temporal cortex. This pathway is important and specialized for identifying and recognizing objects, so they call it the what pathway. Whereas the dorsal stream, which is a dorsal pathway going to the parietal cortex, is called the how pathway. It's sometimes referred to as the action pathway, and it's important for visually guided movements. So as an example, right, I can look at my water bottle. I have my water bottle sitting on my desk, right? If I'm looking at this and I need to identify what that object is, that's cells that are in the ventral stream going the what pathway. What is that object? And if I want to reach out to grab it, to take a sip of it, that's activation in the dorsal pathway, or the how, the how, or the sometimes called where or action pathway. Again, important for visually guided movements. And we can see through studies of people who have damage to these pathways. Again, if there's damage to the ventral stream, I wouldn't be able to identify my water bottle. I might be able to see its component parts, maybe talk about how angled something is, but to put those features together and say that's a water bottle, I'd be incapable of doing that. If I had damage to my dorsal stream, I would be able to tell you that that was a water bottle, but if I went to go reach for it, I would miss. I wouldn't be able to properly guide my movements, nor make the correct grip to grasp that bottle. So again, here are your streams. Dorsal stream, right, to the parietal cortex, guiding movements, ventral stream, the what. I have to label and identify what this object is. Right, so I was just talking about this damage. And, you know, clearly, I hope clearly, normal behavior makes use of both of these pathways in collaboration, right? I need to be able to identify that object and its features to make the appropriate hand grip, to locate where it is in space and be able to extend my limbs accurately in the right place to be able to grasp that object. So it's a collaboration of these areas that inform our actions and our perceptions. I say this a lot in my sensory psychology class. A perception is in service to action, okay? We have to be able to perceive the environmentally, biologically relevant information in order for us to act on it to survive and to reproduce. Okay. So again, receptive fields become larger and more specialized as we move from simple cells to complex cells and then to other brain areas. The inferior temporal cortex contains cells that respond selectively to complex shapes. So not just bars of light, but now we're talking about forms and shapes. Right, cells in the cortex are going to respond to identifiable objects. So I just want to come back here and show you that this is the inferior temporal cortex, and we are going to see more specific feature detectors in that area. Okay, so cells in this area, we've looked at cellular response or timing amount of firing in the cortex of mac monkeys. And what we've seen is that if we put an electrode in a cell in this area of a monkey cortex, and we show them different shapes, we can look at how much that cell responds to the stimulus. And there's there's little response in circles or rectangles, but the more shape becomes hand-like, the more these neurons are going to fire. And so these cells are responsive to body parts, particular body parts. There are also cells in this area that have sensitivity to faces, right? And again, depending on the orientation of the face, you know, or if it's just the back of the head, these cells will change their pattern of response or their firing rate. The less face-like it is, the less activation in these cells. Right, so I said before, the brain is modular. there are brain structures that process information about specific stimuli, right? So we have part of our brain that sees shape, another sees color, location, another perceives movement, another likes body parts, another likes spaces, and we have cells that really like buildings. But those cells are actually space. It's about navigating through space, but they do respond to buildings because buildings are an object that has a deep relationship to space and navigation. So, research that exemplifies that we have areas for processing faces, places, and bodies comes from fMRI studies, and they put individuals in fMRI scanners and they show them series of pictures and they look at activation in different areas of the brain, right? They don't do the microstimulation or putting in electrodes like they do with monkeys. In some cases they may do that, but this is for humans, we put them in fMRI machines. And when you do your chapter 5 reading, you will be more familiar what this equipment does. I've talked about it a couple of times already though. So we have an area in the inferior temporal cortex called the fusiform face area, and that area responds best to faces. The parahippocampal place area responds to spatial layouts and buildings. And then we have the extrastriate body area, which responds best to pictures of full bodies or body parts, like the monkey looking at the hand. Now, what's really cool is how early face recognition occurs. It's very soon after birth. Newborns show strong preferences for right side up faces. If you show infants, newborns, different kinds of stimuli and the amount that they look at that different stimuli, they have preferential attention to faces, which is an indication that they can actually perceive faces. Faces are high contrast stimuli or objects. So newborns without well-developed visual systems can see some, can detect them. Right. Here we see preferences for upright faces as opposed to upside down faces, but they're still looking attention to both. Top heavy configurations get more attention than bottom heavy configurations, right? Because this is more face-like than this. And we can compare an upright face to a top heavy configuration. And what we see here is that, newborns do pay attention to the template of a face, more so to right side up and top heavy objects than they do to upside down or bottom heavy objects. So seems like they have a template for faces at birth. This is very similar to what I talked about, I think, the first week in class when we talked about birds gaping, and that researchers would put a stick figure in front of the bird, and it had to have the appropriate shape of mom in order for the bird to expend resources to gape. If it was a rectangle stick figure, the chick would not gape. It's a waste of resources. And it was evidence that that birds have templates for form especially of mom, and infants have a template for other faces, which could indicate caregiver, right? Caregivers, important for adaptation and survival. Facial recognition continues to develop up into adolescence, or our ability to recognize faces. I have found it really interesting, the relationship between my MS, which attacks my optic nerves and limits my contrast sensitivity, actually makes it harder for me to see faces and recognize faces, and that has impaired my ability to recognize people and remember names and just have more information to put with faces. Quite interesting. Movement, just like other modules, movement, or I should say just like other behavior and phenomenon, movement is not just processed in one area. There is the MTV5 module, which responds to a stimulus moving in a particular direction, but there's also the MST that responds to expansion, contraction, rotation of visual stimulus, or to a visual stimulus. And they both receive input from the magnocellular pathway, right? So the MST and the MT are both modules, but in order for us to have our perception of movement, that's a phenomenon that shows distributed representation, has to be through activation of multiple areas of the brain, or multiple modules that are activated. Right? Stimuli out in the world is too complex for it to be one cell or one area, right? It's a combination of activity in areas distributed across the brain or throughout the brain. Okay, disorders of visual perception. Object, agnosia, IT cortex damage, inferior temporal cortex damage. Here you can't identify objects. We talked about that before. The what pathway is damaged. If the fusiform face area is damaged, people cannot identify faces. It's called prosopagnosia, and it's it's even people can't recognize their own faces. There's a really cool YouTube video that might bring you a little bit into the experience of somebody with prosopagnosia or face blindness. Blind sight. Another YouTube video there. Blind sight is another interesting phenomenon. Let's say you have a participant who doesn't perceive anything, right? Their perception is that they're blind, and you can put them in front of a screen and you can flash light at different areas of the screen, and you ask these individuals to tell you where that flash of light was located based on quadrant, and they'll tell you that they can't see it, and you can press them, just make a guess, your best guess, and they actually can reliably, so better than chance, guess where the light was flashed in these different quadrants. So their perception is actually that they are blind, but there's still information processing happening in the receptors and at different levels of the cortex. So blind sight is a result of damage to V1. However, the superior colliculus is receiving information, and again, individuals can react unconsciously to stimuli without actually seeing them. Okay? So Perception, again, is the actual experience of something. It's the seeing of something, whereas sensation is the detection or the receptor activation. Okay, I put in my slide visual word form area, which I don't think is in your text, so don't worry about it. But there is an area of our brain that responds to words, to letters, to shapes of letters, to written words. And if there's damage in this area, one would not be able to respond to written words. Color agnosia. Or cerebral achromatopsia is the loss of the ability to perceive colors, and it is commonly the result of damage to area V4. What I think is fascinating and don't know enough information about is that even people who have damage to area V4 can still dream in color, and if they've had the experience, they've had the experience of seeing color. And they can also imagine color. And if damage is extended outside of that area, one could lose the ability to imagine color. So visual imagery and actual perception, there are different neurons that respond. We even have neurons that respond to real motion versus illusions of motion. Okay. Another disorder of visual perception is what's called neglect. Sometimes you'll hear it called hemi-neglect. Posterior parietal cortex damage is responsible for this. It seems to be an issue with attending to objects in space. They actually ignore half of their visual environment. They report having normal vision, but let's say you ask a patient who has this type of damage to draw a clock, right? They will put all of the numbers in the clock on only one portion of it. They completely ignore that this exists. Now, if you move the position of this clock to a different area of their visual field, they'll fill in this section and completely ignore another portion of their visual field. Motion blindness, echinotopsia, is the inability to determine the direction, speed, and whether an object is moving. It's likely caused by damage in area MT or V5. Some people are blind except for the ability to detect which direction something is moving. This is probably because MT gets some visual input despite significant damage to V1. Another thing that was, really interesting about my experience, my first symptom with MS was blindness in my left eye. It first started with contrast, losing contrast, losing the boundaries of objects, and then color, and then complete loss of my ability to see form. But I would stand on the subway platform and I could still see fine out of my right eye. It was blindness in my left eye. I would close my right eye when I would hear the train coming, and I could not see the form of the train, but I could see like waves, like moving past my vision. So I could see motion, but had no perception of form. And that was like a felt experience of the modularity of the brain, which was totally mind-blowing. At the same time, it was, horrifying, but it was really cool to be able to have this bit of knowledge and have that experience and be able to really feel the phenomenon that we're learning about. Pretty cool. Okay, so binding, back to binding, right? So again, the brain combines information from different areas so that we can have an integrated experience, right? Distributed representation. It's not just modularity. These areas are active in conjunction with one another. It allows us to form a single percept, right? We can actually perceive objects and separate those objects from backgrounds, and this, depth to our experience. So we're not quite sure how to solve the binding problem. It seems to be related to synchronicity, right, of firing, right, similar patterns in these different areas. But it also might involve the superior temporal gyrus, which receives input from both of those streams, the dorsal and ventral. The parietal cortex, here, damage causes people to neglect. This might play a role in binding. And prefrontal areas. This is where the streams merge and decisions can be made, further and deeper processing of the visual information. Okay, I think that's it. Yeah. Okay, so we are done with vision. I'm going to get this up on Moodle for you today, and I would expect a hearing lecture sometime next week. So enjoy. Take care.